In this video, we discuss about the viroids. So, what is viroids? So, generally, viroids are the smallest known pathogens which causes disease in plants. So, viroids are the plant pathogens. Okay. First, we know the discovery of the viroids. Discovery of the viroids. So, in 1971, potato spindle tuber viroid. This one is the viroid. Potato spindle tuber viroid discovered in potato. Okay, so this is the discovery of the viroid. So first viroid, potato spindle tuber viroid, which is discovered in the infected potato. Okay, nearly 30 viroids have been discovered. Okay, right. So the range in length from 120 to 475 nucleotides. So this is the length between 120 to 475 nucleotides. So they have length between 120 to 475 nucleotides. Okay, right. So what is the difference between the virus and viroids? There is a simple difference between the virus and viroids for better understanding. Okay, for better understanding, there is a simple difference. Once we see, virus means, so this is the genetic material, single-stranded RNA. So single-stranded RNA is the genetic material of virus, and the genetic material is protected by a protein capsid. So this structure is called as virus. Yes, I think you know. Now, what is viroid? So viroid means without capsid, a naked single-stranded circular RNA is called viroid. So that means, this one is the circular single-stranded RNA is the genetic material without capsid. There is naked one. There is no capsid. So that is a simple difference between the virus and viroids. So this one is the virus and this one is the viroid. There is no capsid. That is the difference. Okay, right. So, but the viroids are not produced proteins, but they disrupt protein synthesis in the host. Okay, so generally viroids don't code for proteins. Viroids are don't code for proteins, but they disrupt to protein synthesis. Okay, right. This is the nature of the viroids. Okay, so now we know the mechanism of the viroids. So when it enters into the host cell, what happens? Yes. So here see the diagram about mechanism which is undergone inside of the host cell. Generally plant pathogen viroids, if it enter into the plant. Okay, so plant cell. So this one is the viroid enter into the host. This is the host cell, plant cell. Okay, so it enter into the host cell. So if it enter into when it enter into the host cell, then it goes to chloroplast or it enter into the nucleus of the cell. Okay, if suppose if it enter into the nucleus, okay, so when it enter into the nucleus, it multiply rolling circular mechanism. So multiply by rolling circular mechanism, that is a rolling circular mechanism. It rolled, okay, rolling circular mechanism, then produced long polymeric polymeric strand, long polymeric strand. Then the long polymeric strand cut into monomeric strand, So these are the monomeric strands. Okay, the polymeric strand is produced by the rolling mechanism. Then the long polymeric strand convert into monomeric strand, cut into many pieces. Okay, many copies. Okay, right. By using one host enzyme, that is called RNA polymerase. This is the host enzyme. RNA polymerase is the host enzyme. By using this enzyme, it cut into many monomeric strands. So long polymer strand cut into monomeric strand. Okay. Next, then these monomeric bits or strands pieces comes into cytoplasm of the host cell. Then they are joined each other to form a circular, become circular viroids, okay, right, progenies. So become circular viroids by using ligase enzyme. Okay, so ligase joins the bits and forms a circular viroid. So these circular viroids now it cut through plasma desmata. Here there is a plasma desmata. So move cut through plasma desmata. They cut through plasma desmata and attack a fresh cell. Okay, this is the mechanism.